This method can be used not only to address key questions in a basic research, but also contribute to the medical, pharmaceutical, and biotechnological fields by delineating novel small molecule ligands of essential proteins.
The main advantage of this technique is that it enables parallel analysis of protein and metabolite partners of the protein of choice in close to in vivo conditions. This method helps to fish out interacting molecules without being restricted to pre-selected ligands.
Though this method was developed for plants, it can also be applied for other systems such as HeLa cells, E. coli, and S. cerevisiae.
To begin, prepare MSMO medium and adjust the pH to 5.7 with 1 molar potassium hydroxide. Then, autoclave the solution for 15 minutes at 121 degrees Celsius. Add the supplements to the medium just before the start of the experiment. Now, grow transgenic PSBL Arabidopsis thaliana cell culture in 50 milliliters of MSMO medium in a 100 milliliter flask. Then, place the flask on an orbital platform shaker and gently agitate the cells at 130 rpm at 20 degrees Celsius in the light. Every 7 days, subculture the cells in fresh medium at a 1 to 10 dilution. Next, use a glass funnel with a nylon mesh fitted on a conical flask connected to a vacuum pump to harvest the cells during the logarithmic growth phase. Then, wrap the dried cells in aluminum foil and freeze in liquid nitrogen. Using a pre-chilled mixer